Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Today we're going to look at a paper titled Evidence for Recycling of Synaptic Vesicle Membrane During Transmitter Release at the Frog Neuromuscular Junction. It's written by John Heuser and Thomas Reese. It was published in the Journal of Cell Biology in 1973. We said that a typical nerve cell contains a cell body which has a nucleus, which holds the DNA, contains the DNA. It has dendrites. They're kind of like tree branches, like this, that interface by synapsing with upstream neurons in specialized sensory cells. This part of the neuron, the trunk of the tree, is the axon, and the axon propagates an electrical action potential if the nerve cell is depolarized and reaches threshold. Then there's an all or none response, and the action potential is propagated in this direction from the cell body towards the cell terminus. And then these termini of the neuron either synapse with other downstream nerve cells, or they synapse with muscle at the neuromuscular junction. So here's my schematic of the muscle, and right here is the neuromuscular junction, the connection between the nerve cell and the muscle cell. In this paper, it's this connection, the neuromuscular junction, that Heuser and Reese are looking at. And if you look at figure 1, they show a light micrograph, so taken with a light microscope, and it's a section through muscle, and you can see these pieces of muscle like this. Something like this, and you can see the ends of the neurons here that connect, that synapse with the muscle, and they've circled the neuromuscular junctions in their figure. Like this. So it's a nice figure that just shows this synapse between nerve cell and muscle cell. Now, if you look at this same structure in an electron microscope, this is figure 1 of the paper, and you look at figure 2, this is an electron micrograph, so it's under much higher magnification than figure 1. This is figure 2. And what you can see is a high magnification of the presynaptic terminal of the squid neuron, and then the muscle, something like this. So this is the muscle, and this is the synapse, or the presynaptic terminal of the neuron. So it's just this little point right here where the nerve cell connects to the muscle cell. And within this presynaptic terminal, you can see these pretty dark circles, which are mitochondria. And then you see many, many little tiny vesicles, which are synaptic vesicles. And these synaptic vesicles were shown to contain neurotransmitter. And the idea is that these synaptic vesicles might fuse with the cell membrane and then release the contents, release the neurotransmitter to the synaptic cleft, this little gap between, in this case, the nerve cell and a muscle cell, or it could be between one neuron and a second neuron, the presynaptic neuron and the postsynaptic neuron. But in both cases, whether this presynaptic terminal is synapsed to muscle or whether it's synapsed to another nerve cell, the presynaptic terminal releases neurotransmitter, which diffuses through this little gap called the synaptic cleft, and it binds to receptors that are in the membrane of the downstream cell, the muscle or the nerve cell, and that triggers a reaction in the downstream cell. And in a downstream neuron, that might produce an action potential, and in the muscle, it produces a contraction. So when this nerve cell fires, it causes the muscle to contract. Now, first off, you can't see the rest of the nerve cell here because it's just a thin section. So you don't see the rest of the cell. You just see the presynaptic terminal. Okay, let me try this schematic one more time. So we have our nerve cell with our nucleus and cell body. We have the dendrites, which connect to upstream nerve cells or specialized sensory cells, which are modified nerve cells, specialized nerve cells. And if this fires, electricity travels down to the end of the cell here. And this is hard to see the end here because I'm drawing a low magnification rendering. But if we looked at it at higher magnification, then you would see this structure like this, and that's what contains the synaptic vesicles like this, and then, muscle as we've have it here in the figure. And the idea is that the electricity travels all the way to the end, and we know from Milady and the Adler paper that calcium is involved in this process. And what we know now to be true is that there are voltage-sensitive calcium channels in the membrane, and when the electricity hits the end of this cell, the electricity is sensed, the voltage is sensed by this channel, and it opens up and it lets calcium in from the external media. And somehow the flow of calcium into this presynaptic terminal caused these synaptic vesicles to fuse with the cell membrane. So if I try to draw that again, you have the terminal here, and see if I can draw it something like this. And you have these synaptic vesicles, and this is one of the vesicles coming to the membrane and then fusing with it. The membrane of the synaptic vesicle and the membrane of the cell are the same thing. It's both lipid bilayer, so this fusion event can happen. And then if there's neurotransmitter inside the synaptic vesicle, in the process of fusing, it can be released to the outside of the cell. So a further, image of the fusion might be something like this, and then ultimately the synaptic vessel is completely fused with the membrane, but the membrane, in here in this region is originally membrane from the synaptic vesicle. So that's the idea. The question is, if this happened continually, which it does every time the neuron's fired, then these synaptic vesicles would just continue to fuse with this membrane, and the cell membrane would just keep getting bigger and bigger and bigger because you're adding membrane through the fusion of the synaptic vesicles with the cell membrane. So how is that possible? How does the cell maintain its size and not just continue to increase in size and volume? Heuser and Reese want to study this process of synaptic vesicle fusion, and they want to know where the membrane goes throughout the entire process of neurotransmitter release and recovery. In figure 3, they show an even higher mag image of the synapse between the nerve cell and the muscle at the neuromuscular junction. So they show you a presynaptic terminal. So it's really the neuron is like this, and the muscle is like this. And at higher magnification, they're looking at a region like this in figure 3. You can just see a little bit of the muscle, and most of this top area here is within the presynaptic terminal. So you have a photograph like this, and then you have some muscle down here, and then you have the presynaptic terminal like this, and you can see many synaptic vesicles. Now this is a nerve cell at rest, at the resting potential. So it's not conducting an electrical action potential, and things are just sitting there. And what you notice are you do see some mitochondria, those are the darker structures, and then you see an awful lot of synaptic vesicles. So this is at rest. Then they decide that they're going to stimulate the neuron by putting an electrode in the neuron, in the axon, and stimulating it over and over again. They say that they're stimulating it at 10 hertz. So it's over and over again, pop, 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 and they're stimulating the nerve cell. The nerve cell is conducting an action potential, and it's releasing neurotransmitters, and the vesicles are fusing with the cell membrane. So over and over again at a rate of 10 hertz. If you look at the next figure, which is figure 4, they show another neuron that was stimulated for 15 minutes. So after 15 minutes, we see a big difference in what the presynaptic terminal looks like. Again, we have some muscle down here, and then this region up here is the presynaptic terminal. And one thing that we notice is that they have these new sort of amoeba shaped structures, kind of like this. And they call these cisternae. They name them cisternae. And they're these bigger, weirdly shaped structures. They have these new weird coated vesicles that look like this, and they look like they have little teeth sticking out of them, like this. And you can see them because they point an arrow at these. So these are coated vesicles, and these structures here are cisternae. And then there's fewer synaptic vesicles. I mean, there's still many in there, but they're fewer. So this is after 15 minutes of stimulation. 15 minutes, and again, they're just firing the neuron over and over again at 10 hertz, pop, 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 pop. And the vesicles are fusing with the cell membrane. And then after they stimulate it for 15 minutes, or they take material that's been at rest, they dropped it in a vial of fixative. They immersion, right? We talked about this before. So you drop the material in here, and it gets fixed, and everything gets stuck in place where it is at the time that it's dropped into the liquid. And then this material is processed for electron microscopy, as we've talked about before. So it ultimately gets stained a few times, dehydrated in ethanol, and then embedded in an epoxy type of resin, sectioned to very thin 70 nanometer thin sections, nanometer thin sections, and looked at in the electron microscope. But at rest, we see many synaptic vesicles, and after 15 minutes of stimulation, we see fewer synaptic vesicles. We've seen these weird shaped membrane-bound structures called cisternae, and then you see these new coated vesicles. So in table 1, they show a time series. They show first a neuron at rest. So they have a neuron here like this, and it has an electrode in it, but they're not stimulating it. So this is at rest. And when you look at that, as we talked about before, you see lots and lots of synaptic vesicles. Okay. And then we said at 15 minutes of stimulation, pop, 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 over and over again, then you see some synaptic vesicles, but there are fewer, and then you have these cisternae that show up, and then you have these coated vesicles, the little guys that have these T's on them. And what Table 1 shows is a mathematical summary of measurements of the surface area of all of these structures. So they measure this line here that's the cell membrane. They measure the membrane occupied by synaptic vesicles. Down here, they measure membrane occupied by the cisternae and also by these coated vesicles. So they have three columns in Table 1. They have measurements of a neuron at rest. They have one that's been stimulated for 1 minute, and they have a neuron that's been stimulated for 15 minutes, and then processed for electron microscopy, like we said. And they record the measurements of membrane. So the first they show is the synaptic vesicle amount, and it's very high at rest. There's 330, is what they say. And after 1 minute of stimulation, it goes down quite a bit to 219. And then if you stimulate for 15 minutes, it goes all the way down to 130. So as you stimulate this system, well, in this 15 minute, if you stimulate for 1 minute or 15 minutes, the number of synaptic vesicles, or the volume of membrane occupied by the synaptic vesicle fraction, it goes down. It's high at rest, and then it decreases and decreases more after 15 minutes. Then they talk about the amount of membrane in the cisternae, in these structures. And at rest, there's very little. They don't see very many cisternae. They say the amount of membrane is 23 units. After 1 minute of stimulation, you see 57 units, and then 171. So synaptic vesicles go down over this time period, and cisternae goes up. Then they have coated vesicles, CV I'm going to call it, and there are very few coated vesicles at rest. It goes up to 5.5 units, and then all the way up to 8 units after 15 minutes of stimulation. And then if you look at the plasma membrane, the size occupied, or the amount of membrane occupied by the plasma membrane, you start out at 246. Then as synaptic vesicles fuse with the membrane after 1 minute of stimulation, you have 292, and then 302 after 15 minutes of stimulation. So this suggests that when you stimulate the nerve cell here, that the vesicles are fusing with the cell membrane. I've sort of drawn it like this, something like this. And so the number of synaptic vesicles go down, and the size of the plasma membrane goes up from 246 to 292 to 302. And then you start to see cisternae. Actually, only a few are found at rest, and that only marginally increases. I mean, it doubles at 1 minute, but then it really takes a jump by 15 minutes of stimulation, and same thing for synaptic vesicles. Okay, that's an important figure for the paper. So they then look at this phenomena in a little bit more detail, and in figure 12, they show this beautiful electron micrograph of a presynaptic terminal, again, after being stimulated for 1 minute. And they say that you don't detect easily a big decrease in synaptic vesicles. I mean, they mathematically calculated this in table 1, but if you look at this terminal, you can see, first of all, here's the presynaptic terminal. You can see many mitochondria that have these, invaginations, right? And then you can see all these synaptic vesicles. So after 1 minute of stimulation, you don't really notice a big depletion in synaptic vesicles. There's many there, but it turns out that you do see the cisternae like this. And the cisternae are pretty big, and they're internal to the cell pretty far. And they look very carefully at the edge of the cisternae, and what they can see are some of those little T's of the coated vesicles. And they can also see some coated vesicles here in the cytoplasm of the nerve terminal. But this suggested to them that the cisternae were forming by the fusion of these coated vesicles. Then in figure 14, let's see, 14, this one was 12, they show that at very high magnification you can see the edge of here's the figure like this, and if you look at the edge of the presynaptic terminal, you can see an invagination like this happening, and then the little T's of the coated vesicles. Okay, let's draw another figure. Now they carry out a large set of experiments using horseradish peroxidase. Now we've seen this stain before in the discovery of the blood-brain barrier, and the HRP turns black. I mean, it is developed with a second, or reacted with a second substance once the HRP is loaded into the system, and the combination of the HRP and the other chemical cause the HRP to turn black. Now, we're not talking about neurons in the brain. This is at the frog neuromuscular junction, so the HRP can flow around the nerve cell, and particularly around the terminus. So if you have a nerve terminal like this is the neuron, the presynaptic terminal, and you soak it with HRP, then the HRP is all around this nerve cell. And of course there's muscle here because they're looking at the neuromuscular junction like this. But anyway, there's all of this black HRP stain on the outside of the cell, and they say if they let this sit for a little while, then what they see is HRP on the outside of the cell, but they also find HRP inside the coated vesicles. So you have these coated vesicles like this, and these are now black inside because they've taken up HRP. And what that suggested to them is that they're filling with HRP through an endocytotic process. So you have, I'll draw it down here. You have the presynaptic terminal, and then you have an event, an endocytic event, where membrane is pinching off from the nerve terminal, and this has the stain, the HRP, and the HRP, as this vesicle invaginates, can go inside this area, and then maybe these T's are formed on these coated vesicles, that is, they get coated somehow, and then they are internalized into the lumen of the presynaptic terminal. So it's like this. Now, if they try another experiment where they stimulate for 1 minute, then you have a presynaptic terminal, and of course we have synaptic vesicles in here the whole time, but they are not black. They are clear inside. Then, after stimulating for a minute, they find cisternae, and the cisternae have HRP in them. So you had HRP out here, and you had the muscle. This is 1 minute of stimulation. And you have cisternae now with HRP loaded in there. And you have some of these little areas here have the coating. So they believe that what is happening is that there's this endocytic event here where these coated vesicles are formed at the surface of the membrane through an endocytotic process, and they get loaded with HRP while the invagination is happening, and then these become internal like this. Then a number of these loaded coated vesicles loaded with HRP, they fuse together and form these loaded cisternae. These are shown in figures 20 and 21. At this point, there are still many synaptic vesicles, and again, they are clear. The cisternae have areas that have this coating, and they are now filled with HRP, suggesting that loaded coated vesicles that took up HRP through endocytosis are filled, fused to form the cisternae. Now, if you look at another nerve terminal that's been stimulated for 15 minutes, our terminal in our muscle, then you start to see some of the cisternae disappear, but there are still some, and they are loaded with HRP. But now you see synaptic vesicles form, and they're filled with HRP stain, like this, suggesting that these de novo synaptic vesicles are derived by pinching off parts of the cisternae. Now, if you stimulate for 15 minutes, and then you let the cell rest, you end up seeing the presynaptic terminal with synaptic vesicles. The cisternae are gone, but the synaptic vesicles are now loaded with horseradish peroxidase, like this. And now, if in the process of letting it rest, you wash away the HRP that was surrounding the cell, if you now stimulate this neuron again, then these synaptic vesicles fuse with the cell membrane and they release the HRP into the external media or out to the outside of the cell, and it diffuses away. And now, if you let this cell rest again, then you would see first some clathrin-coated vesicles form with no HRP inside. They would be clear inside. Then you would see cisternae, which are clear inside. Then you would see new synaptic vesicles again with no HRP inside. So, let's just make a summary. You have a presynaptic nerve terminal that's synapsing with muscle at the frog neuromuscular junction, and it has many, many, many synaptic vesicles. When you stimulate this nerve cell and an action potential hits the end of the cell, we said that voltage-sensitive calcium channels open and calcium rushes in, and somehow the calcium causes these synaptic vesicles to fuse with the cell membrane, and so the membrane occupied by the cell membrane, the plasma membrane, gets bigger because the synaptic vesicles are fusing with that membrane and contributing to that volume. The amount in the synaptic vesicles is going down because they're now part of the plasma membrane. At some point, clathrin-coated vesicles appear because they are formed through an endocytic process and recoup the membrane at the cell surface, and they fuse to form cisternae, and then the cisternae liberate new synaptic vesicles by the membrane pinching off, and that restocks the synaptic vesicle pool and makes the neuron ready for the next action potential and the next neurotransmission. Okay, everyone, that's it for today. See you next time.